Welcome to BioLens Learning YouTube channel.
The classic method for isolating individual strains of bacteria from a sample is called the streak plate procedure. In this procedure, a researcher begins by sterilizing a wire loop in a flame. To cool the sterilized loop, the researcher touches it to a sterile agar plate. The researcher then dips the loop into a sample containing a mixture of bacteria. Depending on the sample, the loop may pick up hundreds or thousands of bacteria, which are spread back and forth across the surface of the agar. Before continuing to streak the plate, the remaining bacteria on the loop are first killed in the flame. After cooling the sterilized loop, the researcher drags it through the previous path, picking up a small number of bacteria and spreading them into a new area of the plate. After sterilizing and cooling the loop again, the procedure is repeated. With each new path, the loop picks up a smaller number of bacteria and therefore can spread them farther and farther apart. During the incubation, the bacteria multiply and produce colonies. Each colony consists of millions of cells derived from a single parent bacterium. The isolated colonies found in the last streak represent isolated strains. To ensure the strain's purity, the streak plate procedure is usually repeated a few more times, using an isolated colony as the source of starting bacteria.